Have two or more, 2, 3, 4, 5 of these coming together. These are all called the major calyx. And then you got renal pelvis, and I didn't write it on my chart here, but you should also know that that's the ureter down here. By the way, do you know why these are called calyx? Calyx means cut. And you should know that because in AP1, you studied on the surface of all cells, you have that stuff, what is it called? Glycocalyx. Did your professors teach you that? Why is it called a glyco means sugar. So these are sugar molecules that make a cup shape. Then they're for recognition. They are recognizable molecules that other molecules connect to, bind to, on the surface of all your cell membranes. And it's called the glycocalyx. This is why I teach these words, because here it is, come up. It looks like a cup. And the medical significance of that is not very strongly attached. These can easily be ruptured in somebody that has a blocked ureter, like in a kidney stone situation. That can be an emergency situation, and that's why it's good to know this anatomy. So there's the terms on this picture. You got renal artery and renal vein. Even though I circled them, you already know that because we did that in a cat, right? So that should be a gimme. But you got the renal cortex, renal columns, renal pyramids, renal papilla, minor calyx, major calyx, renal pelvis. I didn't circle ureter, but that I know some professors do that. I don't, but some professors do. So here we go. This is the model, by the way. This is the model that's on your final exam, and they'll point to two of these. This region out here is renal cortex. Yes, excellent. Within the renal medulla, this is the
renal pyramid.
Excellent. Or that one, or this one, right? In between the pyramids, this is the
renal column.
Renal column. At the tip of the pyramid is the renal
papilla.
Anyway, you see only one of these.
Minor calyx.
Anyway, you see two or more, like there or here, major calyx. And where they all come together in this funnel.
Renal pelvis.
Here, renal pelvis. And this down here is the ureter.
So the
ureter is the tube that goes from the kidneys down to the urinary bladder. Yes?
So we don't need to worry about the hilum?
No, they won't do that. It's not on the card anywhere, so I know no professor's going to ask you that. Hilum is actually all of this. We use that same term in the lung. As the airways go into the lung and join the neurovascular bundles that go in with it, that's all called the hilum. Okay. I use this as a bonus station sometimes. This is a real human. Oh boy, that model makes that look so neat, doesn't it? Look at those pyramids. It's not so nice and neat, is it? Look how they overlap each other. Yeah, that's why I love this picture. This is a picture of a real human kidney. We actually do have a specimen, but it's so bad that we can't see anything. This one, we can at least see the structures. So you might see this on there if your professor decides to give you a bonus station. So some of my classes are getting this. And so everything's exactly the same. Renal cortex, renal pyramids, renal columns, renal papilla, minor calyx, major calyx, and renal pelvis and ureter. And all of these would be fair game. I would suggest, though, your professor, like myself, is not going to use the one that's already labeled here. So I would not just memorize that C here is the renal pyramid. If I want renal pyramid on my I don't point to that. I'll point to maybe this one or this one. Yeah, we got to. If this is bonus, you got to challenge you a little bit. For renal papilla, which one did they use? Papilla is E. Oh yeah, they use that one. If I want renal papilla, I'm not pointing to that one. I'll point maybe to this one or this one, right? So be prepared for that. In case you get it. I don't know if you're getting this. You have to ask your professors. Some of my classes are getting this because they need an extra station. I don't have my students just standing there doing nothing. We only have 21 stations for this test. It's not like AP1 where every, 25 stations, 2 questions, not in this. We have a lot of stations that have 3 questions at it, so there's only 21 stations. The only way to give another station is to do something like this. Then we got the nephron model and the renal corpuscle model. For your renal corpuscle, here's your list of things you need to know. First, that it is a renal corpuscle, and then you got the afferent arteriole, the glomerulus that's inside, the efferent arteriole, the Bowman's capsule that's surrounding it, and that it is called the renal corpuscle. What's this? Renal corpuscle. What's this? Afferent arteriole. All of this inside here, glomerulus. Efferent arteriole, surrounding the glomerulus, Bowman's capsule. Some professors might call this the glomerular capsule, so I'm warning you about that. It's not really a choice on the test. It's Bowman's capsule. Some professors also will do this one. That is a choice on the test. What is the covering for this? Podocytes. Podocytes literally are Bowman's capsule. The Bowman's capsule makes connections with the glomerulus and coats the inside, coats the glomerulus. So it's the inner lining of Bowman's capsule. That's what makes podocytes. What they did on the model was remove the podocytes from this one so you could see all of those little holes. They're called fenestrations. All right? I have to let you know that because I don't do this. I point to this, I just want glomerulus. Some professors point to this and they want podocyte. Got it?
Yes.
Any questions? Okay. Also on this, we got this is the proximal convoluted tubule. And this up here, distal convoluted tubule. They didn't put that up there just because it looks pretty. It's literally what it does. Okay, in the body, the distal convoluted tubule goes in between the efferent arteriole. I don't know if you got that in your lecture class. You should have. The cells lining this here, by the way, is monitoring the blood pressure here, which is why those patients that you get into your office that have high blood pressure, you don't just assume what's caused. It could be this. It could be this thing saying there's not enough pressure inside here, and so let's release aldosterone. These are the hormones that get released by these cells to stimulate your blood pressure to go up. And that will require a different type of medication. You're not going to give the same medication for that type of patient. That's the importance of this. Why do we need the blood pressure to be higher? The whole purpose here is to filter the blood. It's to make this thing leak. There's no other capillaries in the body that did this, that had the fenestrations, that had the holes and leak like this. Half of the water that's in the blood that comes into this, half of the water is released. But it doesn't stay that way, of course. When we come out here, these will surround the peritubular capillaries, will surround the tubes. And then we reabsorb 97% of the water that leaves here, 97% will be retrieved. Okay? That's why you need to know about this stuff. This is just more pictures of the same.
Is this picture going to be on the back? No? Oh.
This is just a help study.
Okay.
It's a study guide. It's also what we used during COVID, because the exams were given online strictly. So we used this picture instead of pictures of the models, because you didn't have access to the models. Notice how the podocytes cover the entire glomerulus cell. And where did these originate from? They're just, the inside lining of Bowman's capsule. And that's not a new theme. You learned that. You learned that when you remember the pericardial sac? Did your professor describe that? You got this big balloon, take your fist, and put your fist into the balloon. What's this all out here? Parietal pericardium. What's this lining the heart itself? Visceral pericardium. Same thing. It's exactly how it's formed in the embryo. Okay. Then we got the nephron model, and that's the middle one on that green board. So on that, you need to know the renal corpuscles. You need to know the tubes, proximal convoluted tubule, loop of Henle, distal convoluted tubule. And then when that's done, the peritubular capillaries are on there and the collecting duct. In your lecture class, you are going to get the different names of the different segments of the peritubular capillaries, like the vasa recta. We don't get that specific in lab. It's specifically the peritubular capillaries that surround the loop of Henle, or the nephron loop is called vasa recta, but we don't get that specific. If we come to those, we just want peritubular capillaries, all right? So there's this picture with everything all labeled on it, and then there it is, here we go. This is what's on your final exam, this thing right there. So what's on here? These little balls right there. Those are the renal corpuscles. Here's one that has the tube coming off of it. That is the proximal convoluted tubule. Then you got the loop. That is the loop of Henle. Then you've got the distal convoluted tubule, and that goes, these empty into the collecting duct. These capillaries over here, or up here, these are all called peritubular capillaries. Once more, I'm going to do the other side because you'll all get confused when we reverse the order here. Everything's going to be like in the mirror. There is the renal corpuscle. There is the proximal convoluted tubule. There is the loop of Henle. There is the distal convoluted tubule, and there is the collecting duct. So don't memorize this one's on the right, this one's on the left. The proximal convoluted tubule is the one that's coming off the renal corpuscle. The distal convoluted tubule is the one that's emptying into the collecting duct. So keep those straight, because you're definitely going to get one of those on there. And it'll be on that model. This won't be on your test.